Hello everyone. I'm honored to present our work entitled Two Near-Complete Assemblies Reveal R-subgenome Structural and Centromeric Divergence Associated with Reproductive Isolation in Hexaploid Triticale. Allopolyploidization merges divergent genomes into a single nucleus, creating immediate challenges for chromosome pairing, segregation, and long-term genome stabilization. Centromeres ensure accurate chromosome inheritance, yet how centromeres from different parental species become functionally compatible and diversify after polyploidization remains poorly understood. Hexaploid triticale, generated from tetraploid wheat and rye, provides an ideal model to address these questions. Our findings are summarized in 5 key points. First, we generated 2 near-complete genome assemblies of genetically divergent triticale lines. Second, we developed subgenome-specific antibodies revealing dual loading of wheat and rye CENH3 at all functional centromeres. Third, we found the R-subgenome undergoes extensive centromere remodeling during post-polyploid evolution. Fourth, we identified the R-subgenome as the primary source of structural and centromeric divergence. Finally, we linked this divergence to meiotic defects and reproductive isolation. We first generated a near-complete chromosome-scale assembly for the triticale cultivar ZX830, spanning 15.9 gigabases with a scaffold N50 of 755.7 megabases. The high-quality assembly provides a solid foundation for comprehensive centromere analysis across all 21 chromosomes. Using subgenome-specific CENH3 antibodies, we found that functional centromeres are defined by dual loading of wheat and rye-derived CENH3 on evolutionarily divergent parental sequences. Comparative analyses further showed that post-polyploid evolution drives extensive centromere remodeling, including centromere size convergence, repeat turnover, and centromere repositioning. There are several centromeres followed distinct evolutionary trajectories. For example, CEN4b mainly experienced repeat turnover, whereas CEN1b underwent centromere repositioning. These findings demonstrate remarkable plasticity of centromere identity during long-term evolution. To investigate centromere diversity at the population level, we analyzed 99 triticale accessions and established the first pan-centromere landscape. We found that centromere variation represents an additional layer of genomic diversity, with the R-subgenome serving as the principal axis of centromeric diversification during post-polyploid evolution in hexaploid triticale. Among all analyzed accessions, PT displayed the strongest structural and centromeric divergence relative to ZX830, particularly within the R-subgenomes. We therefore generated a near-complete chromosome-scale assembly for PT. Comparative analyses reveal extensive structural divergence with disrupted collinearity and pronounced rearrangements in centromeric and paracentromeric regions of the R-subgenome. To evaluate the biological consequences of this divergence, we analyzed F1 hybrids between PT and ZX830. The hybrids exhibited chromosome missegregation, lagging chromosomes, micronuclei, and reduced pollen fertility. These observations indicate that R-subgenome structural and centromeric divergence is associated with meiotic defects and reproductive isolation. In summary, this study provides valuable genomic resources for investigating centromere evolution in allopolyploids. It demonstrates that centromere function relies on epigenetic compatibility through dual loading of wheat and rye-derived CENH3. Finally, post-polyploid centromere remodeling, particularly in the R-subgenome, contributes to reproductive isolation and provides new insights into genome stabilization during polyploid evolution. Thank you for your attention.